Well, smart words are only so smart, and then they get dumb. Okay, so then we talked about DNA replication. We see the word replica, it means to make a copy of something. Okay, so that's what DNA is going to do here. It only happens in the S phase of interphase. So if you think back to our cell cycle and we had that circle, I don't know if you remember that, Weston. We have that, we have that, we have G1 and S and G2. That's when we're talking about this. Okay, DNA will make a copy during that step. How does it do it? Well, there's kind of two to three steps. First step is we have something called DNA helicase. And this protein is really only got one job, and that is to unwind DNA. So we're going to go from this DNA that's coiled, helicased, and as helicase moves down this, it's going to basically think of like a zipper. We're going to unzip the base pairs from each other. That's what DNA helicase does. So you don't necessarily need to draw it all out, but I would definitely write down that that's its job. Okay? So let's say it's all unzipped. Second step, we've got another molecule that's going to DNA polymerase is going to add new nucleotides to match the old ones. So let me get rid of this. Okay. So let's say here's my polymerase, which is broken and busted. Let's see if I can get a new one. And it's going to kind of read, we're going to go down this strand, and as it reads each base, it's going to match a new base to the old. So if I read an A, I'm going to put a T. Thank you. Thanks, sir. Just take it back.
It's 239.
Yep, I didn't put it down.
Okay.
If she has a problem, stop there.
Okay.
All right, so if I read a C, what would I put? G. Yeah, and so I, oops. And so as I go down this list, new base, new sugar phosphate, new base, new sugar phosphate, new base, new sugar phosphate. And so I'm basically copying to the old. And then I'm going to do the same thing over here, with the only difference being, because it's backwards, I have to go from the bottom up. But I'm going to do basically the same thing. So I read an A, I'm going to match a B, C, G, G, C, T, A. Okay, and if you look at it, I got A, T, A, T, C, G, C, G, G, C, C. It's a copy. But it's not an exact copy, like a brand new copy and an old copy. What I really have is part of it's new and part of it's old. So here's the old strand, but I've got a new strand to match up to the old. So this we got into an interesting discussion of how old is your DNA. Well, technically your DNA is as old as you are, because half of it goes all the way back to the very original. And some of it's always going to be brand new. So that's where the problems are. You're getting older because your DNA is getting older. So then I forget who came up with it, but they said, well, what if DNA didn't get older? Then you wouldn't get older. Well, that gets interesting. Okay. But that's how DNA makes a copy. It basically splits in half. New bases get added onto the old, and then put it back together. You may. All right. What if it makes a mistake? Well, sometimes mistakes happen. Let's say I didn't put the right Didn't put the right letter. Maybe the wrong one snuck in there somehow. Well, the way your cells catch this, we have those checkpoints at different stages of that cycle. And what it could do is actually cut out, let's say we cut out an entire section that has the problem. And we could then go back in and basically make a new section to replace the bad one. It basically cuts out the old sections that are bad, and we can put in a new section. So there are ways to fix problems. Okay, does that make sense? Okay. Let's take a look at the next part. This is what we got into last time. Gets a little different. This is called transcription. Now, transcription is different because I'm making something called RNA, which is kind of like DNA, but it's a little bit different. And the reason we have to make this is because DNA can't leave the nucleus. And that's actually for its own safety. Because if DNA leaves the nucleus, what's that?
I'm playing Fall of the Leader and I'm gonna get a calendar.
Okay. Yeah, you just get a count. And I'm recording this too, so that's kind of fun. Okay, so DNA is kept safe, but that kind of poses a problem because if I'm keeping it safe, then it can't ever leave, and if it can't leave, then it can never control the cell. And so there's a potential problem there. So let's compare these two to see how they're similar but different. DNA is double-stranded. RNA is only one-stranded, so this enables it to fit through the pores. Right? Okay. DNA's got deoxyribose sugar. RNA's got ribose sugar. So there's a difference. The biggest difference, though, is right here. DNA has thymine, T's. RNA does not. Instead, it has what's called uracil. So we're used to A's, C's, G's. Those are in both. Adenine, guanine, cytosine are in both. But DNA has thymine, RNA has uracil. So that's going to play a major role here in a second. And DNA can only stay in the nucleus. It's locked up, but it's being kept safe. RNA can go in the nucleus, it can go in the cytoplasm, and go everywhere it wants to. Okay. All right, so you got up to that point, Weston?
Yeah.
Okay, so let's take a look at a little animation first to kind of see what's happening here. All right, so we're going to start with DNA. I can tell because it's got T's. And this is going to happen inside the nucleus. There's something called a promoter. Promoter is kind of a start signal. It always has TA. We call it the top sequence. And that's where this molecule is going to start. And whoa, we're kind of temporarily popping it open. But we're matching up RNA because it's got U's. So every time, let me show that again. And then we hit the terminator sequence at the bottom, and that'll tell us where to stop. So we're going to have a promoter section where this RNA polymerase is going to latch on, starts reading the code, but if you look, every time I would normally put a T, I'm putting U's. Everything else is exactly the same, but this is one-stranded. This is two. This has U's every place there would have been T's. Okay. All right, here's the descriptions. I like the picture better, but here's the description. So 3 steps. Step 1, that blob, polymerase. But this time it's making RNA. attaches to a gene's promoter. That's going to tell me where do I need to start. I'm only going to read one side of it. I'm not going to read both sides. So it's not going to end up being double-stranded. It's only going to be single-stranded. Okay. Picture, and it's up to you. You don't have to draw it though. So here's our promoter section, the top. We're going to read a gene, which is roughly about 1,000 base pairs. So if I'm RNA polymerase, I'm going to move along this, and as I'm moving, I'm going to crank out an RNA copy. Just remembering, normally I would have had a T there. I don't have T's, I got to put a U. I still have A's, I still have G's, I still have C's. But every time I would normally put a T, I got to put a U.
Why is that? Because they don't have.
Because they don't have T's to even use. But U's are kind of similar, and so it fits uracil. Okay, so I don't have a T, but I've got something really close called a U, and I'm going to put the U in there. Okay. All right, step 2. That's kind of that's the description. RNA polymerase adds RNA bases to match the DNA bases. DNA is going to rezip. Where's the brain?
Good chilling here in your back.
And then RNA is going to leave. Once we create it, we're going to leave it. It's going to carry the message out to the rest of the cell. Okay. So let's do maybe a little, I don't know, let's do a little practice maybe. Do you still need that? Give you a second. Oh, good? Okay, sorry. Just hanging out over here.
Oh, this stupid, very long genetic RNA.
Oh, come on, stop.
It's just the
It's not that bad.
It's just the longest one.
Okay, so let's say I've got a DNA sequence, and I'm not gonna worry about the other half because I don't really need it right now. And I wanted to make an RNA copy. Okay, so let's say I'm RNA polymerase, and I'm going to read this code, rattle off what it would be.
A, G, C, U, A, U, G, C, U, A, G, C.
Man, you are awesome! Okay, so once I have this, it's now going to leave the nucleus. It's carrying some secret message. Here's what DNA wanted me to tell you. Okay, it's kind of the same idea. All right, so once we have that, what are we going to do with it? Well, it's called translation. Let's see if I can get it to move down just a little bit. All right, so I think of this as very much like taking a foreign language. Okay, you have a Spanish paragraph, you have a French paragraph. They say translate it. Well, this is kind of what the cell's going to do. It's going to translate the RNA we just made into something that makes sense, and that's called a protein. So it's kind of like decoding a message. So we're going to take this RNA sequence, we're going to turn it into a protein that the cell can then use, and the way a protein is constructed, it's made of smaller pieces called amino acids. And think of it like a string of pearls or something like that, where I have one and then another one, and they're just attached to each other. And this is all going to happen in the cytoplasm. We've left the safety of the nucleus. We're now out in the wild, and we could get injured. But as long as I have my original DNA, even if something happens to my RNA, I can make a new one. So that's useful. All right, 3 kinds of RNA that we can make. The first one is called the M for messenger, and the reason it's important, it's carrying the instructions, how to make the protein. All right, so there's going to be all sorts of different messages of how to make different kinds of protein. The second kind of RNA is called tRNA. tRNA stands for transfer, and their job is to carry the amino acids. So I think of it kind of a delivery truck. They're taking materials from someplace and delivering them to where they're going to be needed. So we got a messenger carrying instructions, transfers bringing in amino acids, and then third, little r stands for ribosome. This is actually what's going to make the protein. They're going to read the directions and assemble all the pieces together to make the finished product. And all 3 of these, well, all of them are RNA, but each one of them has a different job to play. Okay, let's take a look at this one. Okay, so here's our RNA that we just made. Okay, so this is my messenger, is this big strand. It's like the instructions. Well, that's interesting. Okay, so here's my transfer. He's carrying an amino acid, like a package. Okay. Here's my rRNA. Ribosomal has 2 pieces. Here's 1, here's 2. They come together. Now the reason they come together there is there's a special message. You may want to write this one down. AUG is a special group of 3 bases, and it's the signal to start. This is my start codon. Codon is a group of 3 bases. All right, now the reason that's so special, Weston, is because if there's no AUG, there's never anything that happens, okay? But if there's an AUG, this whole process starts because the ribosome starts to read the code, okay? Here's how it reads it. When we read an AUG, there's a transfer RNA that fits that perfectly. Here's an A matches to a U. U matches to an A. G matches to a C. Okay? And there's only one combination that fits that, and he's carrying what's called methionine, which we usually just call meth, which is totally different.
Not crystal meth.
It's not crystal meth. Okay? We have a big discussion. But methionine is an amino acid. It's always the first one. Every protein in the world. Methionine is the first one. It's special. Now, once I've read that one, I read the next group of 3, which was AAG, which is going to match up to a UUC. But it's bringing a different package. Okay, now watch what's going to happen between these 2. Ooh, they connected. Okay. This one has no cargo. It's been used up, so it's going to get kicked out to go get more methionine. It can only carry one special package. But I've shifted. I've shifted down. So if I see a UUU, it's going to code for a, what's going to code for? What's going to match up to that? AAA. Somewhere out here, There's an
Oh,
AA,
yeah, I've never seen
and
that.
it fits. And these two are going to connect to the next amino acid and kind of repeat this. We're going to shift, we're going to kick it out, we're going to bring in another. So if I have GGC, lo and behold, a CCG matches.
Oh, yeah, I've seen that.
It's bringing a different one. And each time, we're just going to make that chain longer and longer and longer and longer.
What do they even make?
Good so far? Okay. Now it gets weird here because sometimes we're going to read a group of 3 and it doesn't match up to anything. The RF stands for release factor. So if there's no tRNA that will match up to it, the process basically falls apart. Ribosome's done, my protein's done, and this message has been decoded into a protein. That's how it works. Okay. All right.
When you start doing this worksheet, it makes a lot more sense.
It'll make more sense. Yeah.
Replace with hook?
I don't know if I showed this to you guys.
You do not.
All right, so kind of 2 steps. So here's transcription. It now leaves to go to the cytoplasm. Here's my messenger. Here's my ribosome. Here's my amino acids. Okay, we can see that there's A's and U's and G's and C's. And it finds an AUG start, which is going to bring in my tRNA, which is a whole bunch of the same thing, but just in a weird shape. Matches up. Amino acids join together.
Oh no, we didn't see how this did it.
It's the same thing. Repeat. New connection. Repeat. New connection. Repeat. New connection. Repeat. New connection. I think so. What if I hit a stop?
Then it breaks off.
Breaks off. And we have our finished amino acid chain.
Which will turn into a protein and then go to something else, do something else, I think.